So do you want to start us off on?
Yeah, so I guess this is just a couple of sort of points to start the discussion. So obviously we should talk about whatever ideas that you may have for the vision for the next 5 to 10 years that were not covered by these talks. So whatever
we disagree with.
Yeah. And I think we can also have a little bit of discussion about priorities. What are the really essential things that we need to do as a community, and where do we need to be, and what are the things that would be kind of nice but not necessarily so essential? And what would be, especially in the tech development space or resources, what are the kind of things that would be catalysts to accelerate discovery and sort of like unprecedented opportunities there? We're coming into a space in genetics where clinical applications are really taking off, and that probably should not be at the expense of basic research, but what is that kind of, where do we go between these kind of two areas? And then one question that I want to pose to everyone is that what do we need to deliver to the society, and how do we ensure public trust and support in genetic and genomic research? For example, if we don't develop clinical applications that are applicable to all ancestries, this is something that could actually undermine the public support for the entire field, and we need to avoid those kinds of things. And one thing that I want to sort of mention is that I think the idea is that the detailed discussions about the specific projects and the kind of how do we get there should be mostly taking place during in the sessions to follow, not necessarily right now, but we of course should and could discuss especially the key technologies and resources that are game changers. So with that, I think we can open the discussion.
So my name's Evan Eichler. I just want to, I guess, make two comments. The first is probably predictable for me, but I am actually struck that none of you mentioned evolution. One of the grand challenges of the human genome when I signed up for it was to understand the history of every base in our genetic code. And everything we've talked about, everything has been focused strictly on humans. If we don't understand how genomes are organized, structured, and have evolved, we are not going to solve the missing heritability problem, period. We can't just leave the conserved regions of the genome and focus just on those where we have nice chip-seq signals. We need to actually have comprehensive sequencing, not just of more diverse humans, but of really the phylogenetic tree all the way back. And I think, and I don't understand why NHGRI seems to have gone off the rails on this one, but this is one where it seemed like they were going back on 2 years ago or 3 years ago, but now it's all human all the time. We do need an emphasis on other genomes of high quality. Second point, this is related, and I think Heidi, you touched on this, and Judy touched on this as well as Emma, but I think it's critical that we address the question of variability of expressivity. We have highly penetrant alleles that are some of the most penetrant in a gene causal variants. And we are still identifying individuals who basically appear, for all intensive purposes, essentially normal, with odds ratios of genes mutations in the double digits. And I think what we need are two things. One is we need more grid sampling of the human population. While it's great to have ascertainment for disease or diversity, I think it's critical to actually have large-scale sequencing where we actually have phenotypic recontact and association. So the projects that are being done by all of us are a start, but we need much larger. Second point, which I think you really touched on, Heidi, was this idea that we need to centralize the data and we need to make accessible the phenotypic information and the recontactability of those patients. And I think that shouldn't be restricted to a sub-select few. I think it should be made much more open and accessible to really solve questions of really heritability of disease. Anyways, those are my 2 points.
Jay, did you want to respond? I saw you. Oh, I don't know.
Well, should we, Nancy?
We can.
It's a fair point that the evolution was missed, and sorry about that. But
it was missed last time too, by the way. 2 cycles before, we used to talk about it a lot. But
so I think it's I think it's a reasonably compelling point that a relatively modest, given how cheap long-read sequencing is becoming, that a relatively modest investment in that direction would go a long ways towards advancing the ability to interpret human genomes. Not that that should be the only reason to do it. The second point, I'll just use this as an opportunity to respond to one of Judy's comments about model organisms, and I just want to make a distinction that I think there, and this relates to something that Evan said, there's more than one reason to deeply go after model organisms. Of course, one is as models for disease, right? And I get that there are limitations. The second is as models for learning general principles, right? And for things like variable expressivity, that's the kind of thing that you could really go after in a systematic way in a model organism and learn general principles that would then be applicable to humans, where it's much more challenging to study.
Okay, Nancy.
So one of the things I really resonated with as a master challenge is understanding what every gene in as many genomes as we can look at does. I think our understanding of what all of these genes do is so shallow right now, and in many cases probably distorted by historical ways that we named genes a long time ago, where, when we look at what transcripts those genes make, the name of that gene doesn't even look reasonable anymore for what the gene probably does. I think we must do that in order to do many of the things that everybody's talking about. Annotating genes at every level of function, down to what they do in the organism, is absolutely critical to everything that has been discussed that we need to do.
So I'm a complete outsider to this community. I think I submitted a grant to an HGRI 10 years ago and was turned down. So I'm totally happy to be considered a heretic here. But I think that there is something that is really important for this community to do in order to really address the challenges that will come up in the next 10 years. And that really is to abandon the sort of the comfort zone of the genome. And the reason I'm saying this is because I've heard many times people say either that we need to functionalize genes or that we actually need to understand how genomes encode function. I think that this is a little bit like trying to understand how a car works by only studying the electrical circuitry of the car. The cell really has an extremely complex machinery that is not only encoded in the genome, but then is implemented through protein-protein interaction, signal transduction pathways, interaction with other cells, secreted protein, et cetera. And I think unless we really move away from function into mechanism, and mechanism really on a genome-wide scale or proteome-wide scale, it's going to be very difficult to understand how protein can code function. One of the things that we're learning, so my area is in cancer, but I've also collaborated with many people that study genomic phenotype in other diseases. And one of the things that is striking for me is, for instance, when you look at ALS, is to understand the fact that we have known about the SOD1 dismutase gene mutation for 30 years, but we still don't understand exactly how that works. And that function is clearly not in the genome regulation, but it's actually occurring post-translationally. The other one is that most of the phenotypes that we see in cancer that are regulated at genomic level are actually regulated through very extremely complex loops and autoregulatory circuitry that cannot be described in a way that typical geneticists tend to think about the problem as additive phenotypes. So I think that there's a bit of abandoning the comfort zone and thinking about not function but mechanism at the genome-wide scale, and that could be a challenge for which all the things that Jay, for instance, mentioned and other people mentioned, like for instance using perturbational assays in single cells, can actually reveal those elements of the machinery and then allows us to put them together. The final thing I want to say is that I think that is really critical that we actually contextualize regulation. That is, many of the efforts that I've seen, including ENCODE, for instance, think about regulation as if it were a human cell generic process. What are the transcription binding sites of MYC or of FOXM1? we know from developmental phenotypes and many other phenotypes that actually regulation is extremely specific to the particular context we are studying. And that's a way to simplify the problem. So if we know what are the 400 genes that participate in a particular phenotype, now we can actually go and study them mechanistically much more effectively if we have to do this at the genome-wide level.
Could you please identify yourself, and all future speakers, please identify yourself?
Sure. Andrea Califano, Columbia University.
Rick Myers. This is about the problem of we can understand the molecular effects of variants fairly well now. There are a lot of people in this room and on the stage who've helped to make that happen by high throughput methods. But getting to the organism is, and figuring out whether it's affecting the organism is always hard. And Heidi addressed this by talking about big cohorts and sharing the data, but there already is something in place now that, multiple things in place for, at least for the rare diseases, Gene Matcher being one of them, but others, and I know you use them and know about them, but not everybody does. And a huge amount of this comes from not big cohort studies, but from people all over the world studying some rare phenotype. They get it, they might sequence even a few, put it up there. It's a gene that has not been seen before in disease, and we've done this many times, where you end up with dozens of people, sometimes very quickly, and it's a really easy, cheap way to do that, to then say it very likely is the case, and then also, Jay, it gets at gene function too. I mean, you actually know that at least part of the function is that phenotype.
Yeah, just to jump in for a second. So the Gene Matcher system is part of the Matchmaker Exchange, which is perhaps a very good first example of a true federated global network that has genomic data from patients all around the world that is accessible through APIs and doesn't require centralization. So I hope that that's a paradigm for how we think about the future of federating data. At the same time, it's still a low throughput platform. You know, after you get a hit, it then requires email exchange to exchange phenotypic data. And we'll often, match and then say, oh, it doesn't sound like a match, my phenotype's different than yours. But with deep phenotyping or recognition of differential transcripts, there actually probably is multiple things going on. And I think we really have to think about much broader networks that include not only the gene candidate, but the actual variation, and being able to bring in a much larger cohort than just these one-off. If we're going to solve the area of disease that goes beyond the incredibly high penetrant Mendelian diseases, but gets to everything from the oligogenic to the true polygenic and association studies. So great point. There's so many people with hands up. I'm going to go with Richard Gibbs, and then we'll go to the back there.
Thanks. My question really leads off Emma's last point about how much data do we need to drive prediction. And Jay, you gave the nice example of GenScan, which as far as I remember completely went away when we had EST data. And so even though that was, well, right now I think we rely on measurement, not prediction in that sphere. Well, before you respond and say I'm wrong, my question is really how much do we have to measure? And how much can we rely on prediction? And I think we've got to measure a lot more than we're all comfortable with admitting right now before we can do any meaningful prediction. And that's got big ramifications for the program.
So just on the point about GenScan, well, I'm not going to speak to the history because I don't remember that. I was slightly before my time. But so you're probably right. But in my view, the point of the modeling and the beauty of GenScan is not solely just in the ability to annotate. It's not in the ability to annotate for the annotation's sake. It's in the fact that you have a model that can capture the key features, which in turn can be used to basically have a principled understanding of how sequence features underlie a biological process, right? And so the goal of the modeling that I'm hoping for here, that we're hoping for, I think, if I can speak, is that understanding as attained through models that distill it down to a set of features that actually matter.
Mark, and then Eric.
Yeah.
We're all for modelling, but how much data are we really going to need? That was the question I was trying to lead with.
Can you hear me? Yeah. So I think there was recently 2 papers in Cell that really tried to go from GWAS to testing in regulatory regions to testing them and trying to see what their predictive power was. And it was less than 30%. And I think they weren't testing all of them. They were testing, I think they did 79,000 variants. I think we need to be generating millions of these and testing them at major scale, millions or billions. And I think we also need to be testing the entire region. At the minute, we can only synthesize small regions of DNA and functionally test them. So we really need to be able to generate actually what we're trying to look at. So if we're looking at a regulatory element, we need to generate 1 kb regions or whatever the region of the element is, and then be testing a lot at the minute, I think, of the variants that we're finding to see how they impact gene expression and then taking those into the context of the genome. That's my view on it. But maybe.
Before we go to the next question, we've now heard a lot of questions and comments from the senior members of the community, but since we are thinking about the future of our field, I would encourage also the junior investigators in the audience to raise your hands and speak up.
All right. I'm going to go with Mark, who's had his hand up for a while, and then Eric, and then Sharon, and then I don't know your name. And then we got to move over to this side of the room.
That's flattering. Okay, so I just basically wanted to second Evan's and Andrea Califano's comments. I mean, there's been a lot of discussion about making predictive models, but I really want to emphasize Evan's point that, you know, evolution really shapes how we see the genome today. And if you really want a predictive understanding or a real understanding of the genome, you have to think in evolutionary terms. And I think that's just a very important comment. And then in relation to Andrea's comment, I do think since this meeting is about genotype to phenotype, I mean, the hard problem actually is the phenotype. You know what I'm saying? How do we describe that systematically to correlate with genotype? And I think that is out of the comfort zone and what we should be thinking about.
Eric, and then Sharon.
Well, actually, I'll third Evan's comment, and I would say that the Vertebrate Genomes project we're part of will help that initiative, comparative genomics and evolution. I'll further add, what I don't see enough of the conversation here is an investment in trying to further improve genome assembly quality, the data quality. There are lots of gaps and lots of errors in these genomes still, if you produce data that has those gaps and errors, well, that's what's going to happen in your analysis as well, and will make errors in the variation phenotyping mapping. And I don't think NHGRI should leave that up to some other community or companies. I think they should invest themselves in that effort for the next 10 years to get the data right. And I think it's possible.
Sharon, and then.
So since John Stemyanopoulos is not here, I'll comment on something that his father, George, taught me, which is that you can always think about every question with regard to globin. So I think when we're talking about prediction, oh, it's true, he did say that. When you're talking about prediction, you have to realize how difficult it is to predict disease. So just take globin. You could argue that you could maybe predict some aspects of thalassemia the null, but we never would have predicted sickle cell disease, right? The effect of that specific missense allele on, you know, the sickling, and then the impact of that on the capillary, et cetera, and the pain crises. So I do think a couple of people comment on this, but when we do talk about prediction or even testing missense alleles or every missense allele, how we predict them and how complex the organism probably has to be to really understand the effect of that mutation, I think is not simple. And so I think the issue that Judy probably raised of the mouse versus the fly versus zebrafish, I think these are really important issues in how well we're going to be able to predict phenotypes when testing thousands of mutations.
Hi, my name is Karen Miga. I guess I'm the first junior person to talk. I want to really emphasize the buzz of the comfort zone, but for a different reason. It should really bother every single person in this room, and in fact, I think it bothers most people, that 15 years into the Human Genome Project and we'd still have holes. This was brought up by Eric Jarvis, but I want to re-emphasize these aren't small holes. These are holes that are like millions of bases long that we're completely ignoring and moving way, way fast in our vision plan of how to analyze phenotypes and human disease without addressing these massive issues. And I want to push back, in fact, on what Jay said. First, I want to acknowledge I appreciate satellites. I appreciate the fact that telomere to telomere assemblies need to be in our plan. But I want to say we need more than just a dozen genomes. We need to make this the new standard of human genetics and genomics. We need to have phased telomere to telomere human assemblies. We need to really put our guns out and say, how can we make this happen? The technology is reaching the flames. It's time for us to move forward. And I want to push back on what Emma was saying as well. I think it's really exciting that we have the opportunity not only to read, but to write. But I don't want these regions of the genome to fall far behind again, because they're going to be difficult once again to write. And I think that that's something that, if you look at any type of IF data sets, you can see that transcription factors, transcription, specialized high-C spatial maps are taking place in these regions. We're missing the whole world of genome biology just because it's hard. And this should not be part of our vision plan to avoid things that are hard. And then finally, I'm going to end and say, not only are they hard to study, but all of this falls on the laps of the computational biologist. These are hard to assemble. These are hard for new mapping. This is hard for the new reference genome. And so to have dedicated funds to move into a really hardcore computational biology to make sure we reach this level of completion, I think is really important for the vision plan.
Great, thanks. Move to the side. Whoa. Starting in the back and coming forward.
Hey, Erez Aiden. I wanted to comment about kind of a different aspect of what we do, which is the way that we disseminate results. We produce humongous amounts of data, do huge amounts of computation, and if you look at where technology is at right now versus where it was 20 years ago, it's totally transformed. But if you look at how we disseminate results, I think we have pretty fragmentary infrastructure for doing that. I think the notion of being able to, have a paper instead of, sort of 17th century concept, but as a piece of software that's executable, where anyone can test any line in the code, where anyone can look at any bit of data that it depends on. These are actually very doable things. And I think creating the infrastructure to do that will improve reproducibility of things that NHGRI does and things that everybody else does as well. Simple example of that, just riffing on actually some of the last few points, is if you think about the great product of the Human Genome Project, which is the basis of the Institute, which is the Human Genome Reference, which is, I think, very much showing its age. And there's basic things that you would want to do, like, give me a reference quality genome of a thing, a sample, a person that I could then check. So if I have a new technology, a new way of sequencing, a new way of scaffolding, a new way of anything, I could actually check the accuracy really, really easily. And basic pieces of infrastructure like that don't exist. So you kind of imagine you want to have reference genomes associated with reference people who volunteer for the purpose of, being sort of open in that way. And I think thinking carefully about these sorts of things will enable us to dramatically improve the interoperability and reproducibility of our work.
And I will just add that none of the clinical labs in most of the world have implemented the current Human Genome Reference because of infrastructure surrounding that to make it easily usable. So I think that's a really good point. Coming forward in that row, yes.
Hi, Marilyn Ritchie, University of Pennsylvania. I also want to raise a different point, but kind of speaking to the analogy that Emma raised with the book and the reading and writing, which I really liked. This is kind of a hit to the computational biologists as well, of which I am one, so I'm digging on our whole community. But one of the things that I think we've done pretty well is done the work to try to take the human genome sequence and read it base by base, or letter by letter. So certainly there are regions we've missed that we just heard about, but for a lot of regions, we've done well to put them together into letters. And now I would also say into sets of letters or genes, which are making up kind of words. What we have not done well at all is to put those words in context of sentences or paragraphs. And I think I love the idea of doing these perturbations across the genome, kind of base by base to see what happens. But if we don't figure out how to put those bases together computationally to try to infer phenotype, and I don't even mean just disease, even natural variation, this is model systems as well as humans, I think in another 10 years we're going to be talking about, well, now we understand what every letter in the genome does, but we still don't understand how they come together into words. And if we're talking about biology, it doesn't function letter by letter. And even if we take that analogy, we don't read a book letter by letter. We read it paragraph by paragraph or sentence by sentence. And so somehow we need to figure out how those letters come together, whether that's networks or systems or pathways. But if we I don't want to say that we can perturb them network-wide yet. We just don't have the technology, although maybe by adding in small molecules to perturb the whole network or system, that might help. But I would challenge us to keep thinking about how do we put those letters together beyond just one letter at a time or one gene at a time or one word, but how do those words come together in sentences and paragraphs? Because I think that that's how biology is functioning. And if we don't think about how to do that now, in 10 years, we're going to have even more letters and more words that we still don't know how to put together. So I think coming up with algorithms and strategies on the data we already have, while the molecular people move those technologies forward to characterize function, I think it behooves us to do that.
Bing Ren from UCSD. I want to mention two words that are missing in this great vision presentation by our three distinguished speakers. One is protein. I think to understand mechanisms, as we all recognize to be important for understanding meaning of the genome, we have to understand how they are read, which is by proteins and probably link RNAs and other things. And without a systematic, comprehensive effort to study how proteins, how transcription factors read our genome, communicate with the outside environment, and convert that genome sequence into actions, we're not going to be progressing very far. So I think we got to keep in mind that the DNA is being read by DNA binding proteins and many other things, and we have to find a way to study their function at a scale. Second word, key word, is transposable element and repetitive sequences in general. 50% of our genome are in that origin, in that category, and we keep missing that. And now more and more evidence is pointing to their role in disease, in aging, in neurodegenerative disorders. So why do we avoid that? We should address it head on. We should try to develop technologies, try to develop method to read them, to find how they work, how they mess up, and how they are regulated. So I just want to keep it short.
Trey Heidecker, UCSD also. So I think having the pleasure of sitting in the opposite corner of the room, this point is no longer as complete an omission as it was 30 minutes ago. But I'll say it in a different way, which is we heard a lot from this morning's speakers on the need for, I'll call them genetic perturbational maps or functional maps. And I wholeheartedly, for my part, agree with those points. But I think what was clearly missing is the need for structural maps. The genome is a structure. It's, and connecting that to the phenotype, of course, is the functional part. But to think you can connect that structure all the way to, at the nanometer scale of nucleotides, all the way to changes in phenotype at a patient level of meters, without any intervening structural information, I think is simply naive. And so the conversation, I totally agree with points that were made about the need for more data for modeling, but what kinds of data should we be generating? And to understand and interpret the genome, I think you're going to have to talk not just about functional maps, but about structural maps. Certainly, we've already seen great progress in ChIP-seq. That's a functional interaction. But should we be talking about, how cells are organized in tissues as a structure? That's certainly what single-cell RNA-seq is reading out. Should we more focus on how proteins are organized in cells as a structure, or all of the above? And so I think that kind of conversation needs to be had. And again, at this point, it's echoing the comments, I think, of several people in the room.
Thank you, Heidi. To paraphrase an earlier questioner, I think of myself as the second speaker from the younger generation. I think I'd like to follow up on a point made by Rick, actually. There are many levels of phenotype, as we all know, and we're better at explaining phenotype the closer we get to the gene. Medicine is interested in all the levels of phenotype, but it's a necessity for medicine to understand the organismal phenotype. So I think a fair goal by 2029 is to have a phenotype for every gene, an organismal phenotype for every gene in the genome. Currently, we are at about 20%, so we have quite a ways to go. And I would also point out that many genes, about a quarter of the genes of the phenotype-associated genes in OMIM, have multiple discrete phenotypes that a clinical geneticist wouldn't put together as being in the same gene. So it's even a phenotype for every gene is really a simplification, quite a simplification.
Over this way.
I'm going to build off of Dave and Mark's comment about phenotyping. Maybe I'll target this to Jay just for fun. I worry a little bit. We have a CRISPR hammer. We're going to walk around and look for nails. If indeed we do this perturbation experiment of millions, if not billions, do we know what to phenotype? And I worry that our interpretation of gene or genome function will be a direct result of what we phenotype, what we measure. If we don't measure the right thing, we may conclude a different type of functional impact of those perturbations.
So just to respond, and this is to several of the comments. So I 100% agree that predicting an organismal phenotype from a variant is well beyond 10 years. That's not what I meant. And when I specifically put in there proximal molecular effect, because I think that's what we can do. That's a realistic thing to do, and it's a specific thing to do, right? Just the most ambitious I could imagine would be something like, there are these mice that carry chromosome 21, human chromosome 21, right? Or let's step back from that. So you can imagine the same sorts of global single cell profiling, right? This field is going very quickly, of an organism as effectively being the phenotype, right? Molecular, every cell type, right? Throughout an organism, the ability to pick up subtle changes in expression, the ability to pick up subtle changes in cell type composition. So that kind of thing. There are phenotypes that will be accessible that are more sensitive than things we have done and are being measured through technologies that this community is used to, right? Or is well positioned to exploit, right? So I see the point, but I think there are avenues forward that allow one to do this at scale and learn something, right? In a way that will allow one to understand, again, just the proximal biology of how does the locus work, right? Which we still don't understand. If we're not going to get that, then we're not going to get the rest, right?
And I would add to that there is also ability to do large-scale molecular phenotyping in human populations, things like very, very large-scale RNA-seq, attack-seq, whatnot, that can actually get to some of those kind of regulatory networks in cells, which is not an organismal level phenotype, but is still kind of one step from the kind of molecular phenotype of the genome, then a cellular phenotype, and then you can kind of like get to the physiological ones.
I'm going to interrupt for a sec. Since Dave and Eric both just didn't do this, a reminder again to introduce yourself when you start talking. This is particularly for people who are watching on video.
Emer
Okay. Hi, Emer Kenny from Mount Sinai. So I just have two brief comments. One is I would like to second Evan's comment about the importance of understanding the evolutionary context of every single variant. Two, which I haven't yet heard mentioned today, but I assume that's going to be part of the conversation during this meeting, is really thinking about the move of genomics into lots of different spheres in research and society, and how we're going to tackle the balance between basic research and the kinds of applications that we're starting to talk about this morning. Who we need to engage as part of that conversation beyond our community and beyond the folks sitting here this morning. I think that those are some of the accelerators that we can start to think about as part of this meeting.
Ali Morsavi, UC Irvine.
I wanted to sort of push back on the idea that having the single cell data is going to solve our problems, because my sense is, for example, you know that you've got the single cell data for C. elegans, an organism that we've studied at single cell level for a long time. I'm not sure that the single cell RNA-seq data really added to our understanding of C. elegans development in a substantial way. And I believe that we'll have all the single cell data that we want 10 years from now. And I worry that we'll still not be able to put everything back together, that the real problem is going to be trying to understand how to handle that complexity. The example I'll give you is that right now, the complexity of the transcriptome is exploding. When you think that GenScan solved the problem, in fact, GenScan gave us a very, very simplistic view of what gene models look like. And for the rest of the community, that was fine. They don't want to think about the complexity of the transcriptome or all of the other splice isoforms. But in fact, the number of transcripts that we're going to have in the next few years is going to explode. And we've got a situation where we know that we can generate the data. We can generate the data, we can generate the perturbations and so on, and I'm not sure that we have the computational tools to put everything together, to leverage it in more than the crudest possible way, which is to ignore a lot of the complexity and hope that we're getting 80 to 90% of the picture. So we should be thinking about, yes, we'll get all the data that we need, we'll maybe get even more, we'll get all the perturbations we want, but how are we going to be able to analyze that? Because I don't think the current machine learning tools are going to pull it off for us.
Mike,
Can I
and
just
then go ahead.
Just response to that, I completely agree that cataloging of single cells is not going to get to the answer. We've got to start thinking about the next step of how do you explicitly model the cell-cell interactions, how do you model proliferation, differentiation, clonality. 100% agree.
Yep. Mike, and then we'll go down that row.
Mike Zodi, New York Genome Center. So I want to go back to the very first thing that Jay talked about, which is high-quality genomes and a lot more genomes. And I think not just in this discussion, but for the last 15 years, we've separated those into two different things, that they've been a part of our community working on improving the quality of the genome and a part of our community doing high-throughput genomics. And too often, we're not talking to each other. And I think regardless of what we decide in this room, if I look through my crystal ball 10 years forward, the technology may be that the high-throughput genome is the high-quality genome. But if those communities are not talking to each other, and I note even in the breakout sessions we have today that there's a how many genomes do we need session and a structural variation session, and we're keeping that split. And we need to bring those two together. And I want to tie in also what Heidi was saying at the end of her comments about clinical utility, which is that in large part, it seems like things have moved sort of down the gradient of research, that when it becomes cheap and high throughput in research, it moves to the clinic. And I'm actually not sure that's always the best technology that the clinic needs. Maybe what the clinic needs is actually to jump ahead to the thing that's cutting edge and really expensive. And I think we need to remember that when we talk about moving to the world of the clinic, you know, spending $10,000 for a research subject is really expensive, but $10,000 is basically what we each pay for our health insurance every year. So spending that on genomics for a person in a clinic, if it's actually going to be useful, that's pretty trivial. And maybe we should be thinking about what is the technology we want to drive to the clinic rather than what's the technology we're developing for research. And then let's move that to the clinic after we're satisfied with it.
Yeah, good point.
Hi, Howard Ching from Stanford. So first, I want to sort of echo my excitement with Jay and Emma about perturbing and writing the genome with single-cell resolution. But second, I want to make a pitch for really continue to understand the basic output from the genome. Let's say when we think about the RNA world. Because I think what we think we know is really based on a small set of techniques and the assumptions that those techniques have. And so when we think that we have, let's say, a model of all the genes, things that look like messenger RNAs that we know about, later on we realize there are many long non-coding RNAs, and then microRNAs, things that run off the gel, turns out they're important. And then now there are circular RNAs, tens of thousands of these, which actually can be picked up by your poly-A-directed sequencing. So new technologies that re-explore areas that we thought we knew actually could have huge impact and shouldn't be left off the vision statement as if this was already a done deal.
Yeah, I kind of want to add to that it's actually, so long-read sequencing technologies are discussed mostly in the context of genome sequencing, but actually we're now at the point where we can start to sequence the biologically functional units of the transcriptome, which is transcripts. After 10 years of these short Illumina reads is going to be transformative for transcriptomics to get the full transcripts.
Yeah.
Hi, I'm Ting Wang from Washington University, St. Louis. So I just want to make two quick points. One is when we talk about from genotype to phenotype, oftentimes, when you're in a classroom, you see that diagram, you see environment right in between. Even though there's a whole institute devoted to environment, I think we need to consider that in a broader sense as in context. I think at this point we have a lot of principled way of understanding how the genomes work in the context of reading the genome, in the context of disturbing the genome. But I don't feel we have a principled way of defining all the context upon which we should design our experiment and interpret our result. Genetics, one of the principles that I learned about genetics is there's lots of redundancy in it, in different contexts, right? So without a principled way of designing an experiment, considering this different context, I think we're going to miss a lot of things, right? So I think I'm very excited about all the technologies that we're going to apply to these problems, but I think we also want to discuss how to comprehensively define that context. So my second point is about being comprehensive. So this is something Evan Eckler taught me maybe 15 years ago when I was a trainee. I heard him talk, presenting a seminar in the context of completing the human genome. And I want to expand that concept just a little bit here. First, transposable element. I want that term to be heard and discussed many, many times during this 2.5 days. Thank you, Bing, for bringing that out. But also in the context of evolution. So we talk about evolution from a genetics, genome point of view. As we know, that's only part of the picture. We should, the principles of evolution of gene regulatory network is quite different and remarkable from how protein sequences are evolving. So in that context, I think we need to consider, for example, how epigenomes evolve, how to investigate the evolution of gene regulatory network in the context of one-dimensional genome sequences. So I think these are going to be useful for us to have a comprehensive view of how genotype encode phenotype. Thanks.
Thanks. Charles Lee.
Yes, Charles Lee, Jackson Laboratory. So with regard to perturbations in model organisms, I just wanted to make 2 points. So number one, it's really quite interesting how human geneticists, we all know that if you put the same mutation in a different background, it can actually lead to different phenotypes or severities of the phenotypes. But interestingly, a lot of the work that we do in model organisms is restricted to basically one or a few inbred strains. So I would like to advocate that we think as a community about doing more of our work in more diverse genetic backgrounds of model organisms, and that's the first thing. The second thing is phenotyping in model organisms. You've perturb your favorite gene, and then you specifically look at the nose, or you specifically look at the heart, etc. And again, I'd love to see more holistic studies done where you look at the entire organism. I mean, that's why we're doing it in these model organisms, is to look at what other systems are affected. So just those 2 points.
Thanks. All right, we still have a lot of hands up, and we have 6 minutes left, so keep your hand up if you're under 30. Under 35. All right, assistant professor out there. Good point, Sharon. Adjunct. Go for it.
See, you become an adjunct as an inverse of age.
Go ahead.
I'm sorry. Oh, sorry, which...
Ross is goading me to talk. Eric Mendenhall, Hudson Alpha, sorry. This might be a good time to mention how postdocs are getting much longer in genomics research. But also, I wanted to comment on Sharon's point earlier on how everything can be understood through the light of hemoglobin, and kind of contrast this to some of the stuff that's been thrown out there of looking at 1 million enhancers. Do we really need 1 million enhancers, or do we need 10 more hemoglobin loci dissected as well as that and understood from the phenotypes? That's just one point I was kind of goaded into mentioning.
Great, thanks, Eric. All right, John, go ahead.
Yeah. Hi, John Mudgett. And one of the things that we focused on a long time in my years in research, but also working with Colin on the comp in IMPC, is the return on investment. And one of the things over these 2.5 days, and with regard to your fourth bullet there, is how do we explain that we've been monetized and we've moved the needle? And so going back to Eric's talk this morning, the fact that we are not 100%, but 15% means that our efforts have disseminated our value in this research. And so for us to further the funding, for us to maintain that impact, that dissemination, is how do we baseline where we have come from and where we are now, and how do we envision in 10 years how we will have achieved a return on investment, whether that's leverage of extant grants, whether that is insights that can be monetized on human health. But I think it's important for us to think about how we're going to define the return on investment and then metricize that.
Let's take one from the back.
And then you, sorry. Go ahead.
Thanks. So I want to go back to a point that I think is mentioned, Gil Bejeran from Stanford. Something that I think is mentioned again and again and again, but never in a 64-point bullet point, and I think it should be there, and that is computational tool building. So I just want to remind us, nothing in experimental or medical genomics makes sense except in the light of the computational tools you put it in. Nothing. The data is too big. We all know it. So now if you treat computational tool building as the air we breathe, I would say that is not a good analogy. Not putting computational tool building front and center is a bit like sending your army forward without thinking about logistic supply lines. You don't get as far, and you don't fight as well. So I would like to encourage us to think about computational tool building. I want to give a brief, concrete example. One of the speakers...
Brief, because two more...
Very brief. One of the speakers mentioned genomic natural language processing, right? So their viewpoint was, as a biobank manager, I want to see more genomic natural language processing. But I'll tell you, as somebody who's spent time in these trenches now, first of all, biomedical texts are extremely hard. They're harder than almost any other text. They're not of interest to the general natural language processing community. If Twitter or Facebook are texts that are much easier to analyze. So A, if we don't encourage the type of computational tool building we want, it won't be there. And B,
Okay, we're going to move on. Thank you. We're going to move on to Tim, and then Ben, and then that's it. Go ahead.
Tim Reddy, Duke University. So my comments actually build on that in some ways. I think there's a conflict I'm hearing between trying to be comprehensive and also handling all the complexity that we're dealing with. And I think it's pretty clear to me, at least, that complexity is going to win no matter what. There's infinite conditions, infinite time points, infinite organism perturbations. And I think as a field, from a computational point of view, from an experimental point of view, we have to get really comfortable with having very sparse data. And how are we going to leverage our scientific knowledge in the context of sparse data? We're never going to be comprehensive because you can always treat something a little bit of a different way. And I think we should just own that, accept it, and actually lead the way on how we handle sparsity moving forward.
Great. Ben, and then we'll finish up.
Thanks, Heidi. Ben Voigt, University of Pennsylvania. I'll try to be brief. It seems to me there are two other things that are worth mentioning about this conversation. One is specificity, and the other is the crisis we're building for ourselves. So one thing we've learned from a lot of data collection is that biology is really complicated, and there are lots of connections to lots of things, like EQTLs hit multiple genes. There are multiple points of contact to multiple genes in the genome. So one thing can do a lot of different things. So trying to discern the specific meanings of those things from the sea of all the things that could happen that lead to this genotype-phenotype connection that we're so keen on understanding seems like a critical problem. So solving that specificity problem seems really important. The other thing is this crisis we're laying out for ourselves. We've just posited billions of dollars worth of projects of data collection that's deep and detailed. I don't know how the rest of you feel. I feel overwhelmed and drowned in all the things that we have available, even just analyzing human genetics variation with all the computational tools that we've talked about. Every day a new one is developed, reanalyzing a bunch of data different ways. We have thousands of different analyses that are subtly different from each other that all analyze the same data sets. And now we're having this conversation, rightly so, about collecting a diversity of different types of data. I don't know how we're going to be able to process that unless we have really good ways, getting to back another comment that other person said, about integrating, synthesizing that analysis, making it reproducible, robust, and disseminating it widely. If we don't solve that basic challenge, we are doomed in the next 10 years.
On that positive note.
Can I say?
I have a flair for the genetic, what can I say?
Can I just respond to that? Because I think I completely agree with you. But if we don't have the data, will we develop the computational tools? And also, do we need to understand everything comprehensively, or can we get at the low-hanging fruit initially? And so I feel like it's a really valid point, but I feel like we need to do it to get progress.
Yeah, and I don't think those things are mutually exclusive. I think doing one without the other is a recipe for disaster. I agree.
So we agree we might not be doomed.
Okay. Right. So I'd like to thank all the speakers and panel members for a very and the audience for a very lively discussion. Before we go into break, I just want to make mention that we noted an absence in our thank you slides, specifically for the NHGRI communications team, Alvaro Encinas, who is in the back here, making sure that the webcast is going well, who was here, I heard, until 1 a.m. setting up. We thank you very much, and we're sorry that we left your name off of the slide. We're going to take a coffee break now, and we ask that you all come back promptly at 11 a.m., and we're going to start right up into the next section. Thank you.